Yeah, so thanks everyone for joining us for the PHIND seminar series. We have a really exciting talk on identifying microbiome markers of progression of Alzheimer's disease today. And, oh yes, thank you. And I also just wanted to mention that our PHIND, just mark your calendars please for the virtual PHIND symposium, which will take place on March 23rd of next year. So you'll see a variety of speakers, some familiar faces, some new ones, and we're really looking forward to hearing about everyone's efforts, especially during the challenges now. So I think it'll be a lot of uplifting, exciting work to hear about for this. And also we have some interesting upcoming seminars, both in infant epigenetic markers of maternal attachment style, as well as improving abdominal aortic aneurysms and detective screening. So nice variety of speakers, and hope everyone will be able to attend. And so without further ado, I'll hand it over to Gary to introduce our today's speaker.
Thanks, Ryan. So today we're honored to have Bhatt from the Department of Medicine, Hematology, and Genetics, who's an assistant professor here at Stanford, present to us identifying microbiome markers of progression of Alzheimer's disease. Ami's work is in perpetual awe of how simple microbial organisms can perturb complex multicellular eukaryotic organisms like humans. And she's chosen to dedicate her research program to inspecting, characterizing, and dissecting the microbe-human interface. Nowhere is this interaction between hosts and microbes more potentially impactful than in immunocompromised hosts and global settings where infectious and environmental exposures result in drastic and sometimes fatal health consequences. Ami's group identifies problems and questions that arise in the course of routine clinical care, often in collaboration with investigators at Stanford and beyond. The group applies modern genetic, molecular, and computational techniques to seek answers to these questions, better understand host microbe interactions, and decipher how perturbation of these interactions may result in human disease phenotypes. So with that, Ami, thank you for giving us this talk, and we really look forward to your presentation.
All right, thank you very much. It's a pleasure to be here. I hope you guys are all doing well and are keeping safe. I'm going to be presenting work on behalf of both my group and my collaborators group. We work closely on this with Gavin Sherlock, who is a professor in the Department of Genetics. So the title of our talk is Identifying Microbiome Markers of Progression of Alzheimer's Disease. We received a really generous pilot seed grant from PHIND about 2 years ago now, or a year and a half ago. And I'm really excited to be able to report on where we are in developing and applying our refined measurement tools in this regard. So before I get started, I just wanted to share my disclosures. And we'll go ahead and start with the most important slide, which is, of course, a picture of my group. I regret that I didn't put a picture of Gavin in here, but I think almost everybody on the call is probably very familiar with Gavin, who's done some really fantastic work in adaptive laboratory evolution experiments and identified some really kind of core genetic principles that underlie microbial evolution. Specifically, he predominantly works in yeast. This work started when Gavin was taking a sabbatical, and we were fortunate enough to have Gavin join our lab during that period of time. I'll be calling out the people who've actually done the work as we go through the data slides that will follow. I'll just note that I won't have time today to talk about some very exciting but preliminary work that Sumaya Zlitny, who is a very talented postdoc in the lab, has contributed to this project. And she's been leading the effort to develop and apply metabolomics pipelines to be able to measure microbial metabolites in stool samples, as well as medications and their metabolites in the stool. So I really like this comic. It's funny. You have on the left a gut that's saying, look, I know this sounds crazy, baby, but I feel like we have a real connection here. And on the right side, you have a brain saying, they always say, trust your gut's feelings. You know, if we were convening about 10 years ago and talking about the gut-brain axis, people would have thought we were crazy. But now this is obviously at the top of everybody's minds. It's been on the cover of the New Yorker magazine. It's been highlighted in lots of lay press. And we have a sense based on many, many associative studies that there is clearly some sort of connection between what's going on with our gut and what's going on in our brains. Now, how this actually manifests is unclear as of yet. And we think in our lab that that's in part because we don't have the best measurement tools to measure the microbiome. So our goal in this project was to identify microbial strains and microbial proteins that define Alzheimer's disease and other neurodegenerative disease, and figure out which of these strains and proteins may play a role in pathogenesis of the disease. So over the course of the next about 35 minutes, I'm going to talk about a few different topics. Number one, we can cure and treat diseases using black box microbial therapies. Number two, we'll talk about how we can go from metagenomes to genomes, deriving the genomes of specific bacteria within complex microbial samples without the need for culturing those organisms. And we'll talk about how we can apply this for viruses, bacteria, and beyond. In the last section, I'll talk a bit about small proteins. We have been very interested in small proteins that are encoded by microbes, and we've been tended to try and systematically discover these proteins and validate these proteins at the host microbe interface. So we can save lives using black box microbial therapies. I'm guessing everybody on this call has heard at some point about fecal microbiota transplantation. Pictured on the right are actual fecal microbiota transplant capsules, also known as crapsules. These are fecal material. This is fecal material that has been packaged into a pill, and it can be used for the treatment of many diseases, but the only disease for which it is actually approved by the FDA is relapsed refractory Clostridium difficile colitis. While this is incredibly effective in that context, it's good at kind of changing the composition of the gut microbiome. The problem with FMTs is that we don't know what is in these samples. It's actually quite a mystery. Put another way, this is a photomicrograph of a cross-section of a colon. This happens to be a mouse colon, but a human colon would look very similar. This image was taken by Kristen Earle when she was a graduate student in Justin Sonnenberg's lab here at Stanford. What you can see on the bottom right-hand corner is the colon. That's DAPI counterstain nuclei from the colon of the mouse. On the top left-hand side, you can see all of these different bacteria. And of course, in between the two, you have two layers of mucus, which is in green. You have the thin layer and the thick layer of mucus. Fundamentally, our first question was what microbial strains are present in the gut microbiome of individuals who have neurodegenerative diseases and those who do not. Put more simply, we just want to know who's there in the microbiome. By the numbers we know based on 16S sequencing and previous work that each and every one of us has about 1,000 different microbial taxa that are present within our gut microbiomes. But it may surprise you to know that most microbes have yet to be discovered. And I'm not just talking about microbes that live in extreme circumstances, such as the glaciers of Antarctica or underwater sea volcanoes. It turns out that even in our gut microbiomes, we haven't isolated and cultured most of the microbes that actually are present there. Because of that, we would argue that microbiome research is largely reference-free, meaning if we were to do a simple experiment and to take a stool sample, extract DNA, and compare the DNA sequence that we get to stuff that is in the reference database, we'd be missing a lot of stuff. Therefore, exploring fecal samples really is kind of akin to exploring deep space. We are left knowing much less than we think we do. And this is truly a discovery-based science. Now, if we were to try to understand how microbes in our gut impact things like neurodegenerative diseases, you can imagine that it's incredibly important that we start with a clear and comprehensive list of who's there first. Knowing that the reference databases are incomplete, wouldn't it be great if we could just take a sample like this, do a simple experiment like extract DNA and do some computational analyses, and end up with a list of organisms that are present and their genomic sequences. On the left, you can see what I've demonstrated is a list of organisms that are present within the sample. Instead of having names, we actually have genomes of those organisms represented as Circos plots. So our goal is to go from the mixture of microbes on the right to the organized, assembled genomes that are present on the left. For those of you who aren't familiar with Circos plots, I'll just take a moment to describe how we interpret them. On the outer ring is our goal. That is a complete and contiguous genome. In the case of bacteria, those genomes are about 2 to, let's say, 6 megabases in length, and they're usually circular. On the inside track in this darker color is the type of metagenome assembled genome that we can get using traditional approaches. What we want is something that's more like the colored tracks on the outside, which are much more contiguous with fewer breaks. That process of generating genomes from mixtures of genomes or metagenomes is what we are after. And like I said, we want to do this in a culture-free manner. Because as you probably know, for those of you who've done any sort of microbiology whatsoever, you know that what you grow depends on what media you use and what conditions you use. And that means that the process of isolating and culturing organisms from a mixture of organisms is a necessarily biasing step. And we don't end up getting good representation or faithful representation of what's in that sample if we use isolation and culture-based approaches only. So how can we overcome this? Well, we can actually stitch metagenomes together to form genomes. You can find when you do DNA sequencing experiments, you get these short reads. What if we could take those short reads, find areas of overlap between those reads, and kind of daisy chain those together to get a longer contig? Our goal then would be to take those small pieces, those puzzle pieces as they were, and to do de novo assembly of entire genomes. So that's the goal. Can we take the 100 base pair, 150 base pair type sequencing data that we get from typical Illumina shotgun sequencing, and can we generate complete genomes? To do that, we can actually use a couple of different tricks. And I'm going to take you through a toy example here. So in this toy example, we've sequenced a sample that has 2 different organisms in it. We then took those short reads, we overlapped them together, and we grew them out into longer assembled contigs or contiguous sequences. You can see that these contigs are longer than a read length, but they're still not a full genome. We can then take those contigs, and for the purpose of this toy example, I'm going to represent them as a circle. They are not actually circles, but I'm representing each of these longer contigs as a circle. The size of the circle is correlated with the size of the contig. So bigger circle, bigger contig, smaller circle, smaller contig. We can then take these contigs and we can represent them or organize them based on things like GC content and coverage. It turns out that for a given organism within a sample, the percent GC content is pretty well conserved along the length of the genome for a given organism. Similarly, coverage should be pretty well conserved. And what ends up happening is that these contigs then kind of self-assemble into these clusters, where you can see we have this cluster on the top right-hand side of contigs, and we have this cluster on the bottom left-hand side of contigs. Now, this is a metagenome assembled genome. This is a collection of contigs that we would propose belong together in a particular genome. So how does this work in real life? Well, this is a real life example. This happens to be stool samples from 2 different individuals. These are pediatric patients who underwent hematopoietic stem cell transplantation. The kid represented on the left actually had an inflammatory complication of transplant called graft versus host disease. The kid on the left did not have such a complication. We did that same experiment that I just described. We extracted DNA, we sequenced, We generated those contigs, we represented those contigs as circles, and then we organized based on percent GC content on the x-axis and coverage on the y-axis. And what you can see here right away is that the individual on the right has a much more diverse microbiome than the individual on the left. Amazingly, we've not used any reference database to determine this. Furthermore, we can identify these clusters of contigs that go together. I circled that cluster of contigs that I've colored in green here. That is a candidate metagenome assembled genome. So this idea of generating metagenome assembled genomes from mixtures of microbes in human feces has been pursued aggressively. And in a really exciting advance for our field, at the end of 2018 and the beginning of 2019, 3 papers came out that used this approach on thousands of human stool samples. What these papers were able to achieve was the generation of 200,000 new MAGs, or metagenome assembled genomes. So these are candidate novel bacterial genomes from just these 3 papers. Compare this to the 160,000 bacterial genomes that are in RefSeq, or that were in RefSeq, which is the NCBI reference database of bacterial isolate genomes. So with these 3 papers alone, we were basically, as a field, able to double the number of genomes that were out there. So you might say, okay, well, this is cool, but maybe we're just identifying organisms that are on that kind of long tail of distribution. But that's actually, interestingly, not the case. In one of these metagenome assembled genome studies, this was the study by Pasoli et al. That came out in Cell in late 2018, the MAG paper predicted a novel uncultured genus. This genus is represented in red on the phylogenetic tree on the right. They named this genus Cibiobacter based on the institute that they work at, which is called Cibio. This organism is not a rare organism. In fact, it is prevalent in global human gut microbiomes. So it was kind of puzzling that it had been missed to date. You might say, well, maybe it's not very abundant and that's why it was missed and not isolated and cultured. Also not true. This organism was highly abundant in those samples. It was previously undescribed, as I indicated, and it falls somewhere between Ruminococcus and Faecalibacterium, 2 genera that are very well described in human gut microbiomes. So clearly, with this example, you can see that these metagenome assembled genomes provide insight into uncultured organisms, and that this is an important step toward reference-free microbiome research. But of course, there are problems with these MAGs. One of the problems is that these are very highly fragmented and incomplete genomes. This is an example of a MAG that we generated from our own lab. This is a Phascolarctobacterium species. You can see that in this Circos plot, there are a lot of breaks. You'll also notice that there are these light green sections. Now, these light green sections are actually fragments of the genome that weren't in this metagenome assembled genome to begin with. So these were actually left out. So you can imagine that this is not only highly fragmented, but also quite incomplete. To overcome this challenge, former graduate students in the lab, Eli Moss and Alex Bishara, ended up coming up with a way to overcome this challenge. They used a linked read or read cloud sequencing approach. This was a commercially available sequencing approach developed by 10x Genomics. It was originally developed for human genome haplotype phasing, but we borrowed the technology to apply to these samples. In order to apply this, we had to come up with a way to assemble the data that we generated. Alex wrote a computational assembler called Athena that achieves this objective. And what you can see is we achieved what is in the outside ring, this gold ring, a much more contiguous genome that has all of these light colored segments included, and it's in just 2 pieces. So while this read cloud approach was great, and it was a great first step toward achieving this objective of de novo genome assembly from metagenomic samples, there is some bad news. And the bad news was that read clouds assemble some things really poorly. An example of something they assemble really poorly is the organism Prevotella copri. Now, Prevotella copri is a very abundant organism in gut microbiomes. You can see in the stack bar histogram the composition of the microbiome of this individual using 3 different approaches. You can clearly see that this organism is highly, highly abundant. What you will also notice is that when you take a look at how well this organism assembles, organism assembly, how contiguous and complete a genome is measured by this index called N50. You can see the N50, the larger the N50, the better the contiguity. The N50 of the Prevotella reed cloud genome was very poor. When you get a perfectly assembled genome, the N50 equals the length of the genome. And remember, the length of a microbial genome is on the order of 2 to 5 megabases. So to overcome this challenge, Eli Moss decided that we needed to think about other methods. And Eli was a graduate student in the lab at this time. Now, the reason we were so interested in this was in part because Prevotella copri is not only prevalent in human gut microbiomes, but preliminary data suggests that Prevotella copri is much more prevalent and abundant in the gut microbiomes of older individuals and individuals with neurodegenerative diseases. So if we were really going to tackle that question of understanding how the gut microbiome may change in older individuals and in neurodegenerative disease, it was critical that we were able to get good assemblies for this dominant organism within the sample. So what Eli decided to do was to try and pursue an orthogonal method of sequencing. In this particular case, one of the things that we hoped to achieve was the application of long-read sequencing. Eli wanted to use a nanopore. Nanopores are a way to do long-read sequencing. There are 2 main methods that are being used in the field right now. One is nanopore sequencing, the other is PacBio sequencing. But unfortunately, a major hurdle that we had to overcome to begin with was actually getting enough high molecular weight DNA to be able to actually run a nanopore. The amount of DNA that you need for long-read sequencing is about 2 to 3 orders of magnitude higher than what you need for standard short-read sequencing or for the reed cloud sequencing library-based approach. So the first thing Eli did was to develop methods for high-yield, high molecular weight DNA extraction from less than a gram of stool. And so our goal was to be able to get enough DNA. We needed about 100 nanograms to a microgram of DNA to be able to do nanopore sequencing. We wanted to achieve that from 300 milligrams of input stool. This is the method that Eli ended up coming up with. For the purpose of time, I'm not going to go through this. There are a couple of different methods that can be applied well. Once we were able to generate that DNA, we made nanopore libraries and then ran nanopore sequencing. This is an area where Gavin's participation in the work really helped out. Gavin has been a big fan of novel sequencing technologies and is very closely familiar with the kind of niceties of nanopore sequencing. What nanopore sequencing does is as follows. For those of you who've never actually learned how a nanopore works, this is your opportunity. It's actually a very different method than typical Illumina sequencing, which uses the type of chemistry that we're all familiar with from Sanger sequencing. Nanopore sequencing is not a termination, anti-termination based sequencing. Instead, what happens is your DNA is bound by this protein. This protein consists of a pore, which is pictured in blue, and then an ATP-driven pump, which is pictured in green. This pore, which is a bacterial protein, in fact, is embedded within this membrane, which is colored in gray. The DNA is pulled to the pore and then unwound. A single strand of the DNA is pulled through the pore. And then what you can see is that in this narrowest part of the pore, when a sequence is present within it, it impedes the flow of electrons from the top of the membrane to the bottom of the membrane. This impedance of electron flow is measured and is plotted here on this squiggle diagram. That is then converted into a DNA sequence using a machine learning based algorithm. So this is how nanopore sequencing works. Once we generated the nanopore sequencing, we then had to actually pursue an assembly. Eli created a computational workflow that leverages a lot of awesome state-of-the-art long-read assemblers such as Canoe and Fly. This process involves twofold assembly, merging, circularization, and then finally, because Nanopore makes a lot of errors compared to short-read sequencing, we use short-read sequencing data, so traditional Illumina sequencing data, to actually correct errors within our long read assembly. What we were able to generate really surprised even us. Just to orient you here, on the top, you can see now that circles plot, I've kind of plotted it as a line. So I've just opened up that circle. You can see the short read assembly for Privatelli was very fragmented. The read cloud assembly was very fragmented. You can see that the nanopore assembly was actually totally complete. So we were able to generate a full-length Privatelli genome in one piece using this approach, overcoming a really major barrier that we had had in our field before. Furthermore, you remember this novel uncultured genus Cibiobacter that we had identified using this type of MAG approach, or others had identified using this MAG approach. We were able to generate the first complete closed genome of a Cibiobacter species using this approach as well. The nanopore assembly is on the outside track in blue. I'll just call your attention to the inside track here. You can see everywhere where there is a line, there's actually a mobile genetic element. And everywhere where there is a purple block, that's actually a putative phage element. So now that we've developed these approaches, we can measure these organisms that we believe to be more abundant and prevalent in older individuals. And we're really excited to apply this method to samples that we've been concomitantly collecting from the Alzheimer's Disease Research Center in a collaboration with Victor Henderson, Christina Wiskere, and others. Over the course of the last several years, we've been collecting stool samples from these individuals. To date, we have about 125 samples from patients with Alzheimer's disease, Parkinson's disease, mild cognitive impairment, and healthy controls. To our knowledge, this is the largest such collection in the United States. And we've also recently embarked on a multi-institutional collaboration with other ADRCs in different centers. The objective is to actually compile all of our samples and to have a broader microbiome study at multiple sites. Just to show you some data from these particular samples, we performed nanopore sequencing on 10 of the samples that we collected from this ADRC cohort. Over here are the stack bar histograms for these different individuals. So these are 10 different individuals. On the left of each stack bar histogram is the taxonomic distribution of organisms present based on the high molecular weight extraction that we use for the nanopore approach. On the right-hand side of each histogram is the short read taxonomic composition. You can see that our high molecular weight extractions do a pretty good job of recapitulating what we find using our standard short read approaches. And indeed, we find that many of these individuals have quite a high abundance of Privatelli species. By sequencing these samples, we've been able to assemble complete bacterial and archaeal genomes of many more individuals. Outlined in the dotted red line here are genomes of organisms that we were actually able to generate using nanopore sequencing of these particular samples. And so while it is still very early days in terms of being able to identify specific microbial strains that are enriched in Alzheimer's disease patients versus contemporaneous healthy controls, what we can say is that in this first stage of the project, we've gone from looking at these samples and wondering what's in them to achieving that objective of actually having an orderly set of genomes that we can derive from these samples. In the last section, I'm going to turn my attention to something that is more functionally relevant. You know, at the end of the day, we're not just interested in who's there in the microbiome, but also how these microbes impact host health. If there really is gut-brain communication, we've got to ask ourselves, well, how are microbes communicating with humans? While small molecules have been studied to great effect, we don't know as much about how small proteins, other types of macromolecules may be involved in that signaling. So microbes predominantly use small molecules and proteins to communicate within their own species, which we sometimes call quorum sensing, between species, and potentially even between domains of life. So on the right-hand side, you can see I've drawn a bacteria that's producing a compound, be that a small molecule or a protein. In this toy example, this protein or small molecule binds to a receptor on a human cell and leads to downstream signaling. Now we know that a handful of small proteins are known to play critical biological functions that range from quorum sensing to spore formation to even doing things like regulating these very complex molecular machines as pictured on the right, like these multi-drug efflux pumps. In this particular complicated multi-drug efflux pump, there's a tiny little protein in light blue over here on the bottom left that actually regulates the whole complex. Despite that, the fact that these short proteins do very interesting things, we only know about the function of tens of short proteins in bacteria. And that's in part because these have been discovered by serendipity and they've been characterized over long periods of time. Part of the reason this has been so challenging is that we face a couple of problems when it comes to small proteins. The first is that small proteins are almost never annotated in microbial genomes. That's because you can often end up with spurious small protein calls simply because you end up randomly with a start codon and a stop codon very close to one another. So for that reason, if you take a look at the annotations of these genomes, small open reading frames are almost never annotated. Another problem is that small proteins are hard to detect experimentally. This is because they run off gels and columns due to their small size. And also, if we were to use high throughput methods like proteomics to try and measure these proteins, the likelihood of detection is proportional to the size. So small proteins are much less likely to be detected than large proteins. To overcome this challenge, Hila Sparrow, a former postdoctoral fellow in the lab, undertook a really massive effort to use comparative genomics to annotate these small open reading frames in human-associated microbes. This work was published in the middle of last year. And just to summarize what we were able to find, we discovered 4,500 new families of small proteins using this approach. Most of these proteins are of unknown function. And we predict based on genomic neighborhood, meaning what genes are nearby these small proteins, that about 30% are involved in cell-cell communication. We're particularly interested to dig into those proteins that may be involved in microbe-host communication, as you can imagine. While this was an important step forward for us, of course, we're left with this problem that these are predicted small proteins. And Brian Freeman, a graduate student in the lab, was really interested in how we might be able to ask whether these proteins are really transcribed and translated. Put another way, can we validate these small open reading frame predictions? Now, there are challenges in this process. You'll recall that it would be nice to be able to apply proteomics, but we know that proteomics struggles to identify small proteins. It also struggles in complex mixtures. In a typical great proteomic experiment, we might be able to measure 1,000 proteins or 2,000 proteins if we're really lucky. But in a typical gut microbiome, we covered the fact that there are about 1,000 different organisms. If each of those 1,000 different organisms encodes about 5,000 different open reading frames or potential proteins, that's a total of 5 million proteins that could be transcribed and translated. If we're only measuring the top 1,000 or 2,000, you can imagine that we're missing most of the picture with this approach. An alternative is to use an approach like ribosome profiling. Now, ribosome profiling, which measures protein synthesis, could be a great option. The problem here is that Riboseq, or ribosome profiling on bacterial samples, typically requires that you take a bacterial isolate, you grow up that bacterial isolate in a culture to a 400 milliliter saturated culture, and then you proceed with the traditional Riboseq protocol. Now, that of course is not something we can manage with a fecal sample, and so we needed to come up with ways to get around this. Brain was able to do so by miniaturizing that process, and what resulted was as follows. This is kind of the general approach to Riboseq, and I just wanted to make sure that I got us all onto the same page and explained how this works. This is a picture of a ribosome. As you know, a ribosome, as it translates a messenger RNA, as pictured here, into a peptide, as pictured in these balls, it moves the messenger RNA between the large and the small subunit of the ribosome. What you can do is stall that ribosome, You then expose this mixture to nucleases such as micrococcal nuclease. Micrococcal nucleus or MNase can cleave all free DNA and RNA. Then what you're left with is only the sequences that are protected by being bound, for example, by the ribosome, are actually still present. You can then deprotect those small RNA fragments. You can reverse transcribe them into cDNA, and you can make a sequencing library and sequence. And that's how you end up with a bunch of reads. And so this is how traditional Riboseq works. We're basically sequencing all of the RNAs that are actively being transcribed and only the part of the RNA that is actually within the ribosome. So as I mentioned, after a lot of hard work, Brian developed a method that enabled us to perform ribosome profiling on, instead of 400 milliliters of a saturated culture, on 300 milligrams of input stool. To test whether this method, which we called MetaRiboseq, works well, we compared MetaRiboseq to traditional gold standard Riboseq on mock mixtures. So this is a mock mixture of Staph aureus, B. subtilis, and E. coli, where we've grown up those samples, as I mentioned, to a saturated culture. We performed traditional Riboseq on the mixture. We compared the signal that we get to MetaRiboseq. Each of these dots represents a different gene from the organism indicated by the color, and the log 10 RPKM of the signal of either Riboseq or MetaRiboseq. You can see that we get very good correlation between the two methods, suggesting that MetaRiboseq validates well against the gold standard Riboseq. Furthermore, we wanted to see if MetaRiboseq signal is also, as we would expect, enriched for signal within the coding sequence, so between the start and the stop codon, as opposed to outside of that sequence. And indeed, we find that is the case. As we expect, we also find that we have an enrichment of Riboseq signal at the start and the stop codons. What we then went on to do was to perform MetaRiboseq on various signals, including in our pilot experiments, an ADRC stool sample. What you can see here in these stack bar histograms is 5 different samples, A, B, C, D, and E. D is one of our Alzheimer's disease samples. On the far left column, you can see the DNA metagenomic sequencing and the taxonomic classification of who's there using the DNA. In the next 2, you see duplicates of the RNA-seq data. And in the far right, you can see duplicates of the Riboseq data. And what we find is that MetaRiboseq works well on these samples. Now, I mentioned to you earlier that part of our objective in developing MetaRiboseq was to be able to validate or ask whether these small open reading frames that we predicted were truly being transcribed and translated. And actually, in just those experiments that I showed you in the previous slide, MetaRiboseq was able to validate over 1,000 different small open reading frame predictions, 600 of which were from the subset of 4,500 new families we had predicted in Gila's work, and 400 that actually had been rejected by Gila's very conservative pipeline. So what have I told you over the last about 35 minutes? We can cure and treat diseases using black box microbial therapies, but now we can actually understand who's there. We do this by doing culture-free discovery methods, generating genomes from metagenomes. And I described 2 different methods that we've used to achieve this objective, the read cloud method, and more recently, the nanopore-based method. I highlighted the fact that using this nanopore-based method, we've been able to assemble the genomes of organisms that are prevalent in older individuals. And we hope that this will form the basis of enabling us to understand the strain level composition of microbes within individuals. Finally, I ended with a section that described how we systematically discovered small proteins in the human microbiome and how we've developed methods that enable us to validate that these small proteins are not only encoded, but are actually transcribed and translated at the host microbe interface, getting us even closer to the molecules that may actually be mediating this gut-brain communication. So we end by coming back to this cute cartoon. We now can measure microbial strains and microbial proteins with unprecedented resolution. We also, over the course of the past years, have accumulated a very large pilot sample set of patients with Alzheimer's disease, Parkinson's disease, mild cognitive impairment, as well as healthy control. And at this point, we are poised to define Alzheimer's disease related microbes and microbial proteins and figure out how they might play a role in the pathogenesis of the disease. So with that, I want to thank my lab for their amazing participation and collaboration on this project. I'd especially like to thank Gavin Sherlock, who has been a critical collaborator in this work, as well as Victor Henderson, Tony Wiskare, Christina Wiskare, Nusha Askari, and Katya Andreessen from the ADRC, who have been our partners in collecting these samples, as well as related metadata. I'd like to thank all of our various funding sources, most especially the PHIND funding resource that has enabled us to carry out the work that I've described today. And with that, I'm happy to take any questions. Thank you very much for your attention.
Excellent. Thanks for a great talk and really exciting number of approaches. Yeah, so everyone please, as you're already doing, go ahead and write your questions into the Q&A, or feel free to raise your hand and we'll go ahead and call on you. Looks like we already have a couple questions in the Q&A. One was, have you seen changes in microbiomes in the same Alzheimer's patient over time?
Yeah, great question. So to date, we have not evaluated longitudinal samples yet, but that's actually one of the powers of collaborating with the ADRC. They are following individuals over time. And so we're in the process of trying to collect second and third samples from subjects. One of the big strengths of working within the context of these larger studies is they also collect all kinds of metadata. So it will be very interesting to evaluate how the microbiome composition changes over time in the context of symptoms that are changing, medications that are changing, et cetera.
Excellent. Another question is, how long are the reads from the Riboseq and how does it help to analyze smaller RNAs?
Yeah, good question. So the reads are typically about the size that the ribosome protects. And interestingly, different organisms have slightly different ribosomes and different ribosome sizes, in part because they can have different accessory ribosomal proteins. So the footprint length, which is what we call it, can range from in the mid-20s to in the mid-30s in length. And that, of course, depends on different organisms. In terms of how does it help to analyze smaller RNAs, in this particular aspect, we're trying to focus on RNAs that are being protected by the ribosome. And that has to be done using small RNA-seq, because traditional RNA-seq, of course, is a much longer sequence. So the idea is to not only have some visual, insight into ribosome-protected RNAs, but there are actually all kinds of other interesting small RNAs that exist in bacteria, most of which we don't know the function of. And that's been an area of kind of academic and intellectual interest of Brian Freeman, who has followed up on this aspect of the study. Still very early days, though.
Excellent. Any other questions for our speaker? I guess I have a question in terms of so a lot of this is really interesting kind of diagnostically, and then kind of thinking about what that would mean and what maybe some downstream interventions and those types of things. But I'm also wondering, are you doing things also to kind of perturb the system, like maybe tip it in the direction that might be beneficial to people?
Yeah, great question. I think that is the ultimate objective, right? To understand how the microbiome impacts, these phenotypes so that we can manipulate the microbiome. One of the things I love about studying the microbiome is I think about it as a potentially modifiable biomarker of disease, right? It's like a biomarker of disease, but then we also have tools in our toolkit that can enable us to switch things around. Tools within our toolkit include antibiotics, probably obviously to many. Diet can be a major modifier of the microbiome, and really nice work has been done here at Stanford by Christopher Gardner, Justin Sonnenberg, Mike Snyder, and others on the role of diet and dietary interventions to modify the microbiome. We've done an experiment where we've looked at trying to modify the microbiome using fibers, so fructooligosaccharides, not in this patient population, but in patients undergoing hematopoietic cell transplantation. Other additional ways to modify the microbiome include, as I mentioned before, dietary changes, but also probiotics. And ultimately, one could even think about a process like fecal microbiota transplantation, although obviously it would be ideal if we better understand what we're putting into patients. A related and potentially interesting note is that FMT is being evaluated experimentally in the setting of autism as a potential therapeutic for autism. And though it is early days, Rosa Kramalchik-Brown at the University of Arizona has actually shown some very interesting positive results where fecal microbiota transplantation is actually able to manipulate the phenotype of autism in children with severe disease.
Do you think the effect of microbiome on Alzheimer's disease is more as causal consequences of the pathology?
Yeah, this is such a good question, which is, is it cause, is it effect? And it is very difficult to know. As many of you know, if you happen to know somebody who has Alzheimer's disease, you know that there are a lot of kind of dietary changes that occur, behavioral changes that occur, and medications that are administered. All of these things can actually cause changes in the microbiome, as can potentially the primary disease. One of the things that would be really interesting to evaluate is whether changes in the microbiome might precede the symptom onset of the disease. And that order of, alterations in the microbiome happening before disease onset might suggest that either that's an early biomarker or that there is some contribution. Obviously, the best experiments to do will be dramatic modifications of the microbiome and then evaluating whether or not symptoms are altered or there is a disease modifying effect of this. But these are pretty grand experiments, still very early days. I think that this is even more critical in the context of Parkinson's disease, where in Parkinson's disease, people are known to have very serious, often gut phenotypes such as constipation.
Excellent. Yeah, so fascinating. So many kind of interconnected processes going on. And I think it inspired another question was, do you think the microbiome can be responsible for some of the neuroinflammatory events we see in Alzheimer's?
Yeah, absolutely. I think, that's one of our leading mechanisms that we'd like to investigate, which is, is the microbiome altering the function, number, type of microglia that are perpetuating the phenotypes that we see and underlying the pathophysiology of the disease. So that is an area of major investigation. We actually have a nice collaboration with Anne Brunet's group, where we're interested in connecting microbiome information now in a mouse model to single cell sequencing in the brain and trying to understand the relationship between the microbiome and neuroinflammation.
Do you adjust the finding for the age of onset of Alzheimer's?
Yeah, so we haven't been adjusting our findings for the age of onset of Alzheimer's disease to date. Right now, our cohort consists predominantly of people who have, older age onset of Alzheimer's disease. This is part of the objective of collaborating with the larger national group, because obviously to do these types of adjustments, we need a large enough cohort. As I mentioned, we have one of the bigger collections of samples in the country, and it's still pretty small. So it's our hope that this kind of national cohort that's being led by Rob Knight's group at UC San Diego will enable us to investigate this a bit further and actually to control for a lot of the confounders that are important to consider.
Yeah, I guess, it's always very informative, having longitudinal data. And I know it's hard to come by a lot of times, especially when you have these kind of deep analyses that go on. I know, just kind of speculating perhaps, how, one of the things, one of the challenges I think you're aware, especially since you've been kind to assist with kind of the Smart Toilet project, for instance, is what sampling frequency do you think is reasonable and useful for this type of work?
Yeah, absolutely. I think it's a balance of convenience and data. I will also say, there is also an element of biobanking where, there are practical aspects of freezer space, et cetera, that come into play. I think you want to match your frequency of sampling with the frequency of phenotypic change, the rate of phenotypic change and the rate of exposure to new agents, drugs, diet, et cetera. In this patient population, I think a reasonable longitudinal sampling frequency that balances kind of practical aspects with having kind of dense enough data would be every 6 months or every year. Obviously, this is a way in which something like the smart toilet or more frequent sampling strategies would be really, really advantageous. Sometimes we joke in our lab that you should never flush any sample away, but obviously to collect with that kind of density would be unfeasible.
Of course. Excellent. Any other questions? All right, well, this has been a fascinating talk and so many interesting future directions, and really looking forward to kind of continue to working with you on a lot of these related projects. So, thanks so much for your time today. And it's been a pleasure having you as part of the PHIND seminar series and as a PHIND investigator as well.
Thank you very much. And thanks for all of the great questions.
All right, thank you. Everyone have a great day.